Okay, go
All
to it.
right. So we're recording. Hello, everyone. I'm Ravit Duncan. I'm faculty at the Graduate School of Education and the School of Environmental Biological Sciences. And Moraima, Matus Nicodemos publishes under Castrofy. So basically, that name um, is one of my wonderful seniors, been working with me for a while now, and her dissertation, which is entitled Investigating Issues in Learning Progressions Research, essentially explores learning progressions, which are developmental models of learning. And she focuses that work on a genetics learning progression and has done some really wonderful work on studying and revising this progression. She's published at least 3 articles and 3 journal articles on in this area, and has a book chapter that is currently in press, hopefully be coming out soon also. She's presented about this work nationally and internationally. It's been really well received. She's also a very active member of the GSE and Rutgers community. She's done substantial service. I think she was, I don't know when we started with the brown bags, but she was a coordinator in 2012, 2013. And I think that may have been one of the first, or maybe the second year that we had the brown bags. So she's a founder of this system. She's also has been doing a lot of mentoring and welcoming of students, PhD students at the GSC. She was a student rep to the Rutgers Diversity Committee, and I worked closely with Dr. Evelyn Ehrenreich and around issue of diversity and establishing the first diversity forum. And as a result of this work, she was invited to the Institute of Teaching and Mentoring in Atlanta, Georgia in 2014. She's taught in the Division of Life Sciences and was the STEM coordinator of STEM programs for the Rutgers Learning Centers. And she continues to contribute to our wonderful program and is a highly valued member of the learning sciences community here at Rutgers and beyond. And today she's going to talk about a study that she's done as part of her dissertation, and that explores how students of different ages, right, different levels of schooling, uh, provide molecular explanations of observable inheritance patterns. There's a bunch of genetics. If you have questions for clarifications, put them in the chat.
In the chat.
Um, and I'm gonna hand it over to Moraima.
Hello, good morning. Thank you for coming to my talk. So as Ravid mentioned, I will be presenting my work on the student's molecular explanation of inheritance patterns. So I will begin my talk by discussing genetics and genetic literacy. I will then define the model of inheritance that we use to identify the student's mechanisms. And I will then discuss also the knowledge resources they use to develop these models. Pretty much, I will be talking about how students of different levels of expertise develop molecular explanations of inheritance patterns after they have status quo instruction. So we didn't develop an instructional intervention. We actually looked at students after they had their regular instruction. So we know that genetics is critical, yet difficult. It's critical because genetic is important for personal and civic engagement. For example, if you are going to make the decision whether GMOs should be sold in supermarkets or whether CRISPR should be used for treatment of some disorders. Also, interpreting results from genetic tests like 23andMe. 23andMe, as we know, they give you your individual probability of having a genetic disease. And we know from the research in education that individuals might have difficulties in understanding what it means to have a gene for something and what is the potential impact that this gene might have in their health. So um, also, like another difficulty with genetics is that schools have a hard time keeping up with the technological advances in the field, and this leads to misunderstandings about these concepts. So the reasons why genetics is difficult is because one, there are multiple explanatory models that are involved in explaining the effect of genes. For example, when experts think of a gene, they think of them as a region of your DNA that code for proteins, while the general public might see these as determinants of who you are. So you have a gene for obesity, or a gene for diabetes, or a gene for whatever thing, right? So they don't see genes as something more than just giving you something, a trait. And this is where genetic determinism comes too, right? So because if you don't understand how these genes are bringing these effects and you see them as something giving you a particular trait, you might believe that the genes just give you that without considering the role of the environment and or other aspects in the cause of disease. For example, if you have diabetes or you have a gene for diabetes, doesn't mean that you're going to get diabetes. It means you have a predisposition for it. But there are certain environmental things that can be done that lead to the onset of this disease. So an issue with this issue of genetic determinism sometimes comes from the examples that we use in instruction that we tend to use reinforce the idea of one gene per trait. So given these difficulties that I just discussed, researchers like Stewart, Cartier, and Passmore have argued that to become literate in genetics, you need to understand the linking between three models of inheritance. The inheritance model, which is here, I hope you can see my cursor. This model explains the patterns of correlation between genes and traits, and it involves the when genes are dominant, genes are recessive, what are the probabilities involved in this. And this is one of the first models people learn in school. The framework also includes the molecular model, which consists of the connection between genes and traits. So pretty much it involves the protein-based mechanisms. How are genes bringing about the traits and how proteins are involved in this process. And the other model that is part of this genetic literacy is the meiotic model, which explains the processes that allow the transfer of genetic information from one generation to the next. So we wanted to find out how students develop molecular explanations for inheritance patterns. How do they reason about these linkages? So one question that is useful to analyze how students link these models is to think about what occurs in an individual that is a heterozygote. By heterozygote, I mean an individual that has one allele coding for one type of protein and another allele coding for a different or mutated protein. Why is it that heterozygotes of sickle cell are healthy, but heterozygotes of Huntington, for example, are affected? Why is it that the same configuration of alleles gives you a different result? In the dominant one, you're affected, but in the recessive one, you're unaffected. So how does this happen? So in the recessive story or the recessive disorder, I'm showing you in the left panel here what happens in the healthy individual, in the individual that has two normal versions of the genetic instructions. So what happens is these two alleles here are going to code for a full amount of the normal hemoglobin, which is the protein in the red blood cells that transports oxygen. And their phenotype is healthy. Their red blood cells are going to look normal, and they are going to have a normal shaped hemoglobin that it's going to be able to transport oxygen. In an individual that has one normal and one mutated version of the genetic instructions, half the amount of the normal hemoglobin is enough to lead to have a healthy phenotype. However, half of their hemoglobin is going to have the abnormal shape and is going to not be able to transport oxygen, and the other half is going to be normal, leading to a red blood cell that has an abnormal shape. In the individual that has two mutated versions of the genetic instruction, all the hemoglobin protein is mutated, and it's going to lead to a phenotype or a trait that is expressed in the body by having two copies that are of the two cells that are abnormal, or all the cells are abnormal, and the hemoglobin is unable to properly transport oxygen like these would. So that's what happens in the recessive. What happened is there was a change in the recipe of the instructions for the protein, and this caused a change in the cells and therefore also a change in the way the organism can transport oxygen, right? The person. In the dominant story, like for example, Huntington disease, you are going to have two normal versions of these genetic instructions and a full amount of the normal Huntington protein. Therefore, their phenotype is having normal neurons. The individual doesn't have any disease. In comparison, in the heterozygote, what I said before, the individual that has one normal and one mutated version of the genetic instructions, half the amount of the protein is going to have abnormal length and the other half is going to have normal length. However, having half the amount of the abnormal length is too much. So it's going to cause you neuronal damage because they are going to be, this protein is going to affect the way the neurons, right? And in the individual that has the two mutated versions of the genetic instruction, there is not going to be any normal length Huntington protein, and that leads to neuronal damage. So what you can see here is that you're going to have the idea that these alleles are coding for a change that is reflected at the cellular organismal level. In the dominant story, one abnormal copy it causes damage, but in the recessive story, half the amount is enough for you to be healthy. So again, a mutated gene creates changes that are reflected at the cellular organismal level. So how do scientists think about this? So generally, there are two ways to think about mutations and the effect in protein function. You think about them like loss of function or a gain of function. In a loss of function, you're going to have a change in the protein that is going to cause a complete loss of the protein function. And in the gain of function, you can have a new function of the protein. The protein is going to do something that makes it different from the normal protein. So if you think about the examples that we talked about before, we are going to have that the dominant story, in this case, this is a gain of function because in Huntington disease, there's neuronal damage caused by that new protein. And in the recessive story, there's a loss of function. So what we wanted to do, we wanted to see if the students could come up with explanations that were similar to what the normative model is saying. So our research questions were, so we have our main research question, which is how students reason about the connection between these two models, the molecular and the Mendelian model. And then we wanted to look at three things. One, what mechanisms do they use and do they relate to the loss of function, gain of function? Two, what knowledge resources do they use to connect these models? What are the things that they allow them, that helps them be able to think about those models that way? And how do these explanations change over the course of schooling, so over time? So this study was a cross-sectional interview study in which I interviewed students across several grades. I interviewed middle school students, graduate students, 11th grade students, AP students, undergraduates, which consisted of some first year biology students and some genetics majors. I also interviewed PhD students. And after they had their status quo instruction, so they all went through their genetics instruction first, and then I did the interviews. I timed them so that it will be after they had their class. So in the first task of my interview, we asked the students how to describe and reason about the inheritance of a recessive disorder using pedigrees. And we also asked them to explain the molecular mechanisms for this disorder. So pretty much what we did, we asked them to explain why Bill here is affected by the disorder. And they had to figure out that it was because their parents must have been a carrier. So I showed you before that this individual here, the heterozygote, carries one healthy and one affected allele. So they had to figure out that this pattern was recessive. And they had to then also explain why. One of the questions I asked was, why are these healthy and why is Bill sick? And they had to, one, explain the inheritance model aspect of it, and then they had to explain what they thought about the molecular reasons for that. And then in the second task, we asked them about a heterozygote, again, an individual that has two alleles, one healthy, one mutated, that had a dominant disorder. So in this case, they had to reason about a dominant disorder and explain what were the reasons why this individual is actually still sick. And we will give them this question. They had to choose. The correct answer was A. And if they gave that correct answer, they had to elaborate on why did they think that that was the correct answer and what were the mechanisms that they thought that were involved there. And if they had the incorrect answer, I would still like ask them to explain why they chose that. In the next question, it was a similar question as Sam. It was also about a dominant disorder, but in this case, the protein is affecting the function. So I wanted to see if they could come up with ideas that are along this area, and they had to elaborate and explain why they believe that this was the correct answer. And the third task was we gave them 16 genetics terminologies, and I will ask them to make connections among them. This is actually an example of one of the ones that links that they built, and they had to explain why they formed those linkages. And I also will ask again questions about, we'll go back to in this part, I will sometimes go back to the first question to elaborate more if I felt like they didn't explain properly what they believed. So their analysis consisted of, I first identified episodes in which the students were explaining the biological basis for inheritance patterns. And then I used the existing research in education to inform the content analysis, to identify the types of mechanisms that they use to link molecular and classical genetics. And we also use a cognitive model of molecular genetics to identify the knowledge resources the students were using to actually support those models. And also we use the normative model of genetics that I talked about earlier, and I'll go over again, to identify the different mechanisms that they were using. So for example, again, this is the normative model. So we focused on a loss of function, gain of function. And also we looked at if they will describe as a reduction of protein or if they will talk about a complete loss. And also we looked if they will talk about an increase in protein function, or if they talked about a protein interfering with another one, or if they mentioned a protein acquiring a new function. So in the normative model, we expected them to put the loss of function with recessive and some dominant, and the gain of function with the dominant. But and this is our model. So we also wanted to look at the knowledge resources they used. And to do that, we used a model of student reasoning that was originally developed by Duncan using students' explanations in molecular genetics. So she interviewed undergraduate students and she asked them questions about molecular genetics. And she found that the students had a particular pattern of reasoning that consisted from a general explanation to a more specific explanation that included domain-specific heuristics, which were key components of the system that illustrated the important relationships between the different levels involved, and explanatory schemas, domain-specific explanatory schemas, sorry, that included the interactions and mechanisms that were possible in the domain. And I'll later on will go explain in more detail like how that pattern of reasoning looks like. But what this model allowed us to use was to, first of all, see if we can expand this model from molecular to classical genetics, but also to have understand what were the domain-specific knowledge resources that students will be using when they were reasoning about these things, right? And then, for example, for the domain-specific heuristics, one example is the gene code for proteins. So we've been talking about it from the start, right? We said that genes determine what proteins you get, and that affects how your phenotype is going to be expressed. So this is an important heuristic, and we wanted to see how students of different grades will use these, but also if we could expand this model to include the inheritance model. So how does this pattern of reason look like? So I asked Michael in this interview, why is Sam sick? So if you remember, Sam is from the second question. He says, Sam has a dominant disorder. His mother's genes are B or B, and Michael's genes are this. That means that the dominant allele is masking the function of the recessive allele. So this is a general explanation that is, he could have answered that without having any genetics. But later on, he elaborates this explanation to include more domain-specific knowledge. And you can see it in the next slide. He uses the heuristic of proteins and centrons. They're functioning normally, so some of them are, and that genes are coding for protein. And then he uses a mechanism of inhibition to talk about what proteins are doing. So we wanted to see what knowledge resources they were doing. And what we found is that for the inheritance, they used a two allele for each trait heuristic, which consisted of understanding that there are two alleles determining traits. And also the idea that for recessive disorders, sometimes one allele is enough for be healthy, but for other disorders, you might have that one is not enough. Also, we identified different explanatory schemas that including a silencing or turning off of a gene, and also one gene interfering with the function of the other. So these were the knowledge resources. What were the mechanisms that they use? So for recessive, the student, okay, so what were the kinds of mechanisms that students were using? So first of all, I'm just gonna very quickly go over what we asked them. So for recessive, so in recessive, the students were trying to answer the question, why are these two individuals not sick when they still have one copy of the gene that is affected? So when we asked the students to elaborate and ask why is this sick and why are they not sick, they not sick, they came up with different explanations. For example, this is an example of a student mechanism. This student is actually I think a high school student, and he said that the recessive allele is being turned off. So he said the allele A, the little allele, is there, but is being turned off by the dominant gene. So maybe the protein binds to it. So what he means with this is that the protein made by the dominant gene is turning off the recessive allele. So pretty much, even if this mechanism is not 100% normative, they are still using ideas that are that are and are being operationalized at the correct level, because they're understanding that there can be genes involved in the inhibition of another one. Also, they use these knowledge resources to be able to think about it. And this is just a small piece of it. I have a longer transcript that shows how he used these ideas too. So then in the mechanism of compensation, so that was another mechanism students use, they said the healthy allele produces enough amount of protein for the A to be masked. Maybe it makes a substantial amount that you can even see the effects of the little a. The protein, the little a codes for. So they're saying that the big allele is able to produce enough. So they understand that the dominant allele can compensate for the recessive. And those were the two types of explanations for the recessive and then for the dominant. And remember, in the dominant disorder, we have that having one mutated allele is enough to be sick. So the kinds of explanation we observed were so haploinsufficiency, one allele doesn't produce enough. So the student says that the father doesn't make any of the protein because he has one dominant allele coding for not producing the protein. So it must mean that you will need more than one allele to make the protein, or one copy allows you to make, but not enough. So they are, and the reason why I call this haploinsufficiency is because that's in the normative model, this is the way that having only one allele not producing enough is called. So they are using this idea, even if they might not know what is the normative name, but they are understanding that one allele is not enough. And then we also observe a dominant negative or interference mechanism that in which they believe that the protein coded by the allele binds, forming dimers and inhibiting the function of the protein. So they understand that one protein can inhibit the function of another one, and these are the knowledge resources they're using to do that. So the third question that we had was like, how do its explanations change over the course of schooling? So how do they do as they are like learning more about genetics? So we found that one, these mechanisms are easier to conceptualize and develop earlier than another. So for example, the compensation mechanism and the haploinsufficiency mechanism, what the students were able to give those kinds of explanation even at the middle school level. It wasn't a lot of the middle school students, but it still shows that students can learn and understand this. They can understand that these mechanisms exist, even though they are saying it in the fly. They still have they can still understand this. So so the reason that we believe that these mechanisms might develop some earlier, some later, some are easier or harder than others, like we think that the dominant is harder, is because of the knowledge resources they have available. So the knowledge resources provide the heuristics and schemas necessary to develop the mechanistic explanations of inheritance patterns. So the use of the two allele heuristic, for example, allows the students to understand that there are two alleles coding for different proteins, right? And that they are two for each trait. And this, it seems really important for the students to be able to understand the mechanisms also. So it seems we observed a shift from explanations from dosage to silencing. And this is very interesting because it shows that we can help students develop these by helping them develop the knowledge resources. So, and this is, I just forgot about that, sorry. So if we go back to our original question, okay, so are students able to think of loss of function or gain of function? So what we found is that they seem to be able to think about loss of function in the right context in recessive. And they are not, they didn't really come up with ideas of increase in protein function or an acquisition of a new function, but they did think about interference for the dominant disorder. So the reason why we think that we might have not observed these two, it could be that our task privilege certain explanations over others. So it could be that if we develop an interview that specifically asks this, we might observe that. So again, absence of evidence is no absence of evidence. It might be that our task privilege a certain explanation over other ones. And then so our findings, we identified new heuristics and schemas that extended the molecular framework into classical genetics or the inheritance model. We identify new ways the students use molecular mechanisms to explain patterns of inheritance. And there seems to be a progression in terms of sophistication. So they shift from early explanations of dosage to gene expression. This was difficult. It was difficult put within reach for high school and middle school students. It seems that the understanding of the molecular basis of dominance is harder than recessive. And we can begin to specify where instruction can focus on helping students develop these knowledge resources. Because we know that an important heuristic is the two-allele heuristic, and also Ron and I and David have showed that in a previous paper in which we did this with middle school kids. So I think that the findings of this work just reiterate that it's a really important idea. And then also it's important to reiterate that recessive alleles are not simply passive entities. They code for proteins that are structurally different, and that these proteins can lead to changes, but however, we don't observe them because of the molecular mechanisms we discussed. And also instructions should include the notion that gene code for protein. That's a really important thing. That's one of the heuristics that Duncan identify, and that these functions can have a direct result in the organism's physical traits. And thank you so much to everyone that helped me with the interviews. They're listed here, or with reading or other help. And thank you for coming. Does anybody have any questions?
Hi, I have a question about back to like the interviews, and I wanted to know because you interviewed so many people, and I'm just thinking about my own dissertation work, and I'm wondering how many of the interviews were face to face, or did you do like a combination of surveys or questionnaires?
They were all
Like
face to
how was it
face.
set up?
I did all this face to face, but I did do some through Skype. Back
Mm-hmm.
then, people use Skype, and I would just record it in my computer. But I did them face to face. But I have people help me also. So Veronica, Nama, we
Mm-hmm.
so so Ravid and I and the other research group, we trained together on how to do these interviews. So we will do a part of them, and then we will look at a video of them after that, so that we ensure that everyone was asking them the same way. I did the majority of them though, because it was my study. So
Right.
they did help me with the AP and the middle school students and the high school, but I did the college and the PhD students.
Okay. Phenomenal work.
Thank you.
Congratulations.
Thank you.
So, Mariah, very interesting talk.
Yes, thank you.
And could you go back to the slide where you compared the results you got from the different groups, the middle school, the high school,
Yeah.
the college, PhD?
This one?
Let's see. I'm not seeing it at the moment.
Oh, okay, you can see it. Let me
Table
just
where
share
you
again. I'm sorry.
compared the results on
Yes.
the various levels of students. Yeah, this one. Um, so since I work with university and PhD students,
Mhm.
I have to say I was struck by the numbers 3 and 4, the interference schema and the silencing schema, and how few of the PhD students got it.
You know what? How I
It was sort of,
explain?
were these biological students?
Yes, yes, they were all from biology, but
That's pretty shocking.
but they had different levels of expertise. So it could be the way that I explain this is that they might, you know, how as people become specialized, they start forgetting other things. So I feel like maybe they were becoming too specialized in what they were doing, and they just didn't talk about this. But they did give good explanations. The thing is that they might have focused in one or the other. So I interviewed, I forgot how many now. So some of them will give both types of explanations, but some will give only one of them, right? So that's why it was 12 of them. So not all 12 of them came up with these, right? And some of them will just stay at the heuristic level, which is also, I mean, it's not a bad explanation. It's just they didn't elaborate it further. You understand? So they might have just stayed at, okay, so one allele is enough, but they wouldn't elaborate. Even if I asked, they wouldn't elaborate what schema was, what mechanism was involved there. You understand? So if they didn't elaborate it, I can't code them that way. So um but I just think that it's because, and that there's other work with PhD or researchers that supports this idea that the more specialized somebody becomes, they start forgetting the little details of other things. So it could be that that's why. Yeah.
Although I have to admit that he's right in that this is not a particularly good picture. And in
Yeah.
other work that we've done and some work that I've done with colleagues that involved undergrads, not necessarily grad students, but undergrads, including senior undergrads, they really don't perform at a level that you would expect
Yes.
a senior undergrad to perform in terms of what they know and the kinds of explanations that they're able to provide. So this is a bit alarming. I think one of the issues that we've seen in the past in the study that I did with the undergraduates is that there's so much detail given in lectures sometimes about specific mechanisms that the forest gets lost for the trees. And they don't get the overall sort of schema and what's the game here, and they get details, but then when you give them a novel phenomenon, which all of these were novel to them in that sense, it wasn't that they learned it in class and had to remember it during the interview. These were novel, and when you give them novel situations, they oftentimes struggle to use these ideas. So they know stories that they've learned in class and can tell you cases of examples from class, but it doesn't materialize into some larger knowledge, more stable knowledge structure that could be used across contexts.
So for example, if you PhD people, for example, will give me an example of, oh, the promoter became inhibited or the promoter was turned off. Well, yeah, but that is still a silencing schema, right? You are turning that off, but they just can't, if you give it to them in another context. So you remember how like we get all these different models of how these things work, but it's hard to, like, sometimes, like David saying, bring it, transfer it to another example if they don't really understand what the underlying schema is, right? So
So it sounds like in some sense what you're saying is that, or one possibility, I don't know if this is what you're saying, but one possibility is that, you know, at the higher levels of knowledge, for instance, they're really looking at mechanistic details.
yes, details
Rather
like
than overall grouping of all these mechanisms lead to gene turnoff because of structural change.
I feel like they need to take it one level up when they're in their instructions, sort of like not just focus on what are the little details that are happening there, but like just what is the global picture. I think that that's the issue. Yeah.
This would be a very interesting study to extend to medical students after their first year and in the middle of their fourth year.
Yeah, that's interesting, right?
They're on the front line, really, of explaining this in the community.
Yeah.
Or medical students and residents, because
so I thought also about people that are doing genetic counseling. Will they answer this differently than like somebody that is actually a researcher, or will they answer it differently as somebody that is like an undergrad, you know, like have a different way of like thinking about it? Yeah.
Very interesting. Just as a comment, I think nice to take this and give this talk or one of your other talks back in Mayaguez.
Yeah,
I can make that connection if you.
Yeah, sure. I'll do it. I love to. Yes. So any other questions?
Anybody else have any questions? Okay, it looks like we'll end a little early today.
Okay. I should have planned it.
Good planning, good planning. Thank you very much, Moraima. It was nice to see you.
Yeah,
thank you for presenting for us.
Nice to see everybody.
Thank you for joining, everyone.
And thank you for coming. We can email Dr. Langer.
Yeah. Thanks for sending me the note.
Of course, yeah.
Have a great
Bye.
today, everyone. See you all next week.
Moraima, you've got some people thanking you on the chat. Just so
oh, okay. Let me see. Thank you. David, we still meet tomorrow, right?
Yeah, I think
yes,
so.
Yes, okay.